Good morning, everybody. Scott and I are going to talk to you about medical research on Alzheimer's disease for about 40 minutes, and then you'll have an opportunity to ask some questions. So let's kick off. Today, I'm going to be asking you for two things by the end of this lecture. I'm going to be asking you for your brains and for your money, because by the end of this talk, I'm going to be telling you about the beginning of a new clinical trial that intends to test what we think might be the first ever cure for Alzheimer's disease. And I use this term very carefully, but I do mean it. So it's time to get a little bit excited in the sense that Melbourne, particularly the Florey, has led the world in pioneering some research about the cause of Alzheimer's disease that has led all the way to a drug candidate that is going to be tested coming up beginning next month. And we're going to tell you about that today, what the basis of it is. You might realize that Alzheimer's disease is incurable, that there are treatments for it, but currently there's nothing that stops the disease in its tracks. So in this realm of neurology, it is one of those areas where we have, unlike other parts of medicine, almost nothing to offer except Band-Aids. And that's very sad. So let me give you some background to the history of all this and tell you how we got to this exciting position. This is one of my favorite illustrations of the process of Alzheimer's disease. It is a series of self-portraits by a famous portraitist called William Utermohlen, as he came down with Alzheimer's disease from the age of 60 when he had his first symptoms down to the age of 66, I think, if I remember. Yeah, 66 by the time he was completely demented. So over a period of 6 years, he went from almost normal function to a person who was ready to be in a nursing home, and subsequently he died. And you can see how his ability to assemble a representation of his inner world disintegrated, and he went from a very powerful portraitist to someone whose just the vestiges of his skill were left. He could still summon a powerful image, but nowhere near as organized. Now that is typical Alzheimer's disease. It's a slow process where all of the things that you've learned, all the skills that you've had slowly evaporate. It is the most common cause of dementia. What does dementia mean? Dementia comes from the same root as the word mental, which means thinking. So dementia, loss of thinking, loss of cognitive function, in particular, memory and reasoning. And Alzheimer's disease is the most common cause of dementia, which accounts for about 70% of cases. And we can divide up the pie in terms of causes of dementia. And here you can see Alzheimer's being the biggest component, and then the next biggest cause of dementia is vascular dementia, and there are a number of other diseases which are less common, such as frontotemporal dementia, Lewy body disease, and even Parkinson's disease can cause dementia. And this is important because when we get down to a clinical trial, as I'm going to tell you about, we actually have to pick the people who have got Alzheimer's disease and not the other causes of dementia. So we can't just take everyone, or else we're going to run out of money, because we've got only a certain amount of money to spend on a clinical trial. It's a lot of money, but we can't afford to test people who are not Alzheimer's disease. It's taken us a long time to be able to make the diagnosis accurately, because in the past, we would have to rely upon a person dying before we could be certain that they've got Alzheimer's disease. Now we have progressed through the work which largely came out of Melbourne and was largely due to the credit of Colin Masters, who has been one of the pioneers in this field in Australia, that we have a method now of making a diagnosis of Alzheimer's disease without having to actually look at the brain itself. And we do that with scans that I'll tell you about as we go along. Here's some handy facts about how big this problem is. Worldwide, there are, now the number's bigger. There's more than 45 million people who have the diagnosis, and this is expected to reach 135 million people by the year 2050. And the risk for dementia or Alzheimer's disease increases with age, especially in retirement years. So that by the age of 85, 1 in 3 people will have dementia. And this is, 85 is no longer regarded as a far-fetched age. In fact, most of the people in this room could reasonably expect to live to the age of 85. By the year 2050, more than 3% of Australia's GDP will be spent on caring for people with Alzheimer's disease. Now, that's a huge number, 3%. The financial cost to Australia currently is in the order of $7 billion a year, and, all right, I mentioned that 3%. So let's get down to the actual symptoms, or the history of it. It was first described in the year 1906 by Alzheimer himself. There's a nice picture of him, and it is called a neurodegenerative disease. That is a group of diseases where the grey matter dies off slowly. And the first symptom is short-term memory loss. So that's why I was asking you the question about short-term memory or memory. If you're worried about your memory and you're seriously worried about your memory, you may be a candidate for the clinical trial that's coming up. It is a gradual process, and eventually all the facets of brain function deteriorate, and there are changes in personality and behavior, and late in the disease there's loss of bodily function. Curiously, it's a disease that doesn't kill you. It kills off the parts of the brain that are needed for higher function, but not for things like breathing and swallowing and even movement, simple movement, so that you can stay alive provided someone feeds you and cares for you. So the average death occurs about 7 to 10 years after the diagnosis is made. This is an old slide. We used to say that it could only be certain with an autopsy or brain biopsy, but in the last 2 years, the consensus is that we can make the diagnosis based upon an amyloid scan that I'm going to show you shortly. And shows you how old this slide is. We also used to think there was no diagnostic or predictive test. Well, I'm very excited today to share this lecture with Dr. Scott Ayton, who is a dementia fellow. So our National Health and Medical Research Council has a program for dementia research where they have given awards to young investigators, and Scott is one of the most promising of that cadre of fellows. And we've been collaborating, and we think we now have a predictive test for Alzheimer's disease. It's the first of its kind. It's going to be published and announced on July the 25th. So the fact that you're here, you get an inside scoop to hear about this. So we think we can now make this prediction. And the diagnosis also, in terms of the clinician, used to be dependent upon the presence of symptoms. So the patient would have to have short-term memory loss and other features before we'd start thinking about whether or not it was Alzheimer's disease. Now, as a practicing clinician, I was taught during my medical school days that if a person turned up, let's say at retirement age, and said that they've got short-term memory problems, they can't remember people's names, phone numbers, they keep losing their keys, I was told to tell them that this is benign senile forgetfulness. Don't worry, take a Bex, lie down. It's not going to be Alzheimer's disease. This concept is gone. Although if you speak to your GP, very often the GP is taught to say this to you. Actually, we do worry about this now. So if a person has got memory loss, we're now just at the cusp of that era where we want to take it far more seriously. And we want to determine whether or not they have Alzheimer's disease, because we're just getting into that era where we are testing drugs which may actually change the course of the disease. Currently, when the practice is that if a person turns up with a dementing illness, you want to treat what you can, because about 5% of cases of dementia are treatable in the sense that we can reverse something that is not Alzheimer's disease. And the causes of those sort of dementias are problems with hormones, infections. So there are things you can treat that cause dementia that we recognize. So anyone who has got a dementing illness gets assessed very carefully by a neurologist or a psychiatrist in order to determine whether or not they've got one of these treatable forms of dementia. But by far, the most common thing is Alzheimer's disease, which is not currently treatable. What we have, when I say treatable, I mean it's incurable. What we have are these Band-Aid type treatments. And these are the list of drugs that we offer patients. They include Aricept, which you may have heard of, Exelon. These are smart drugs, essentially. A normal person who would take Aricept would perform better on an IQ test for a few hours, and that's how they work. It doesn't change the disease. And then there are other drugs like Namenda or Ibixa, which buffer some of the symptoms, but also don't really change the cause of the disease. There are other problems in Alzheimer's disease, such as depression and epilepsy, which are complications of the disease, and these have to be treated. And there is always a big question about what can you do in terms of over-the-counter and prevention and so forth. I won't go into that a lot right now because I want to actually tell you what the basis of the clinical trial is. And of course, there are a lot of treatments that have been frustrating and not worked. And by far, despite many, many clinical trials in Alzheimer's disease with a great number of candidate drugs, nothing has worked. And that's how it has been. It's been a graveyard in terms of failed clinical trials. So what do we know that we can really hang our hat on, so to speak? In Alzheimer's disease, the main pathological feature, the thing that you look at in terms of the brain, is loss of the grey matter. So the meat of the brain disappears gradually. This is a normal brain, and this is a brain with Alzheimer's disease. So a person with Alzheimer's disease will lose about half the mass of the brain by the time that they are demented. But this doesn't happen, as I say, rapidly. It happens very slowly. And in fact, much of that loss can occur without any symptoms actually being present. So most of the time, this is silent. And the question has been, can we detect this early enough to do something about it, to present an intervention, a candidate drug, at a point where it's got the best chance of being able to help? In that regard, and I touched on this earlier, Melbourne has been in the forefront of the diagnostic technology involved, and that is the detection of the invisible amyloid accumulation within the brain that occurs even before the shrinkage of the brain. And what we have there is what's called a positron emission tomography scan for amyloid. What this involves is turning up for a 2-hour session where you have an injection of a very, very small amount of radioactive chemical which gets trapped by the amyloid in the brain. And then we scan it. And if you're on the left, that's normal. If you're on the right, that's going to be Alzheimer's disease if it's not Alzheimer's disease already. And this becomes one of the diagnostic tests for Alzheimer's disease. We can see this process even while a person is functioning completely normally. These are the sort of candidates we think would be best off in terms of treatment. This is a presentation from Europe about the theory of Alzheimer's disease from the microscopic level, which actually, it goes for about 5 minutes. It's very nicely presented, and it's largely wrong. So let's watch it. Beautiful presentation this is.
Alzheimer's disease was described for the first time in 1907 by the German psychiatrist Alois Alzheimer. In performing a histopathologic study of the brain of his patient Auguste D, suffering from dementia, he brought to light the presence of 2 types of lesions in the brain, senile plaques and neurofibrillary tangles. He reached the conclusion of a distinct disease of the cerebral cortex. 100 years later, thanks to current scientific techniques, research has made a great leap in the understanding of the disease. We know that the brain is made up of neurons and that these are interconnected to form a vast network. These connections, known as synapses, enable the transmission of information from one neuron to another. In Alzheimer's disease, 10 to 15 years before the appearance of the symptoms, 2 main lesions form in the brain: senile plaques, composed of amyloid beta protein, and neurofibrillary neurofibrillary tangles composed of tau protein. How is the senile plaque formed? On the surface of the neuron is a large protein called APP. Normally, APP is sectioned by enzymes on the surface of the neuron, and it frees a protein called amyloid beta. The amyloid beta protein is then cleared in the body. In the case of Alzheimer's disease, there is an imbalance. The amyloid beta protein is no longer regulated and is found in too great a quantity. The proteins assemble to form indissoluble fibrils and create senile plaques. How are neurofibrillary tangles formed? When a neuron communicates with another, a signal goes from the body, known as soma, to the synapse to transfer the information. The signal passes through the skeleton of the neuron composed of microtubules. These microtubules are stabilized by normal tau protein. In Alzheimer's disease, tau protein becomes defective and detaches from the microtubules. Thus, the skeleton of the neuron dissociates as it is no longer maintained. Defective tau proteins then assemble to form filaments in the neuron. Without the skeleton, the neurons degenerate and connections between the neurons are lost. The abnormal accumulation of tau filaments in the neuron creates neurofibrillary tangles and eventually causes the death of the neuron. How do the two lesions spread throughout the brain? Neurofibrillary tangles and senile plaques do not follow the same pathway in the brain over time. Neurofibrillary tangles first develop in the region called the hippocampus, which is essential to memory and learning. They then reach the whole brain following a centrifugal movement. The process causes atrophy, which engenders global dysfunction. The progression of the lesions corresponds with the symptoms of the disease, which begin with memory problems, followed by problems of language, recognition, and incapacity to perform gestures. Senile plaques develop differently. They are initially observed in the cortex, secondly in the hippocampus, and then the senile plaques reach the whole brain following a centripetal movement. Their progression does not correspond to the symptoms of the disease. But numerous questions remain unanswered. We know that the presence of the two cerebral lesions is necessary to develop Alzheimer's disease, since one does not come without the other. But which lesion comes first? Neurofibrillary tangle or senile plaque? The answer is still under debate. Many clinical trials destined to reduce senile plaques in the brain have failed. In fact, reducing them is not sufficient to eradicate the disease. It has now been suggested that well before formation of senile plaques, smaller forms of amyloid beta called oligomers appear to be toxic for neurons, disturbing their communication when they fix onto synapses. It would appear that the toxic oligomers and their accumulation in senile plaques are at the origin of neurofibrillary tangles, which in their turn are responsible for symptoms. The relationship between amyloid beta protein and tau protein is still little understood. What is the exact sequence of molecular mechanisms leading to the development of dementia? What is the role of genetics and environmental risk factors in the appearance of the disease? Scientific research is essential in answering these questions. Thanks to researchers, Alzheimer's disease is better and better known in its complexity, and new avenues raise real hope for the eradication of this devastating disease.
It's a nice presentation. So what's wrong with it? What's my quarrel with this? Well, what you've seen there contains quite a bit of dogma. And the main dogma, which is very appealing, is that when you look down the microscope, you see two things that shouldn't be there. You see the plaques and the tangles. And therefore, since the get-go of the research in Alzheimer's disease, the temptation has been to say, well, somehow the plaques or the tangles must be the cause of the disease. But medicine is full of instances where spots appear, and that's not the cause of the disease. Let's consider measles. In measles, you get a skin rash, right? You get these spots, right? But you wouldn't treat it with a cream, correct? And so there are these biases that get introduced into medical research where if you didn't know better, you would treat what you could see. And this is an instance. And as that film alluded to, just targeting that pathology alone, what you could see down the microscope, we actually can get rid of it. We can get rid of the spots, but the disease continues to rage on. So something else is going on. And that's been our philosophy with the research, that it's not as simple as just the plaques or just the tangles. There's something else in there that hasn't been considered. So the work that we've done here has focused on that something else, which brings me to the next slide. So I've been at this for a long time. I actually did my PhD with Colin Masters shortly after he purified the amyloid out of the human brain. So I studied amyloid a lot. But the question always is going to be, what is it about aging that makes it a risk factor for Alzheimer's disease? And so that's where we've come from. That's where we think the best clue is. The main question that should be answered is, what is the chemical change in the brain that occurs with aging that increases the risk of Alzheimer's disease and might act in synergy with that pathology that I mentioned, the plaques and tangles? And so we've hunted for this. And how do you hunt for something like this? Well, you have to study normal people, you have to study normal animals, and you have to study the process of aging at its absolute quintessence. And that's why when we do medical research, sometimes we have to unravel the whole situation down to its really primitive, primordial level. And for that reason, we think about the origin of life itself. And you may or may not know that life on Earth is a consequence of the emergence of metals into the crust of the Earth approximately 3 billion years ago that set up a kind of chemistry of self-replicating molecules that depended upon the availability of iron. And as a result, all life forms on Earth, with very few exceptions, rely upon iron in order to develop, in order to grow from birth. So life depends upon iron. This is like biology reduced to really basics. Life depends upon iron, but here's the interesting thing when you study the biology of iron. There's no pathway for excreting iron. Now that means that if you're a healthy animal or a healthy human being or a worm and you encounter lots of iron, which you think is great for your development, in humans it's needed for the development of the brain, it's needed for the development of blood and so forth. Whenever you encounter it, you gobble it up. And this is all life forms on Earth are like this. But the problem is that because evolution has made us so hungry for iron, we have no means of getting rid of it when it becomes excessive. We just store it, we put it away somewhere and keep it for later on the assumption that all we need to do is reproduce. But of course, as far as humans are concerned, there is this great era after reproductive life where you go and do other things. You have grandchildren, you take up another vocation, you learn. There is this great epoch that is not something that evolution ever considered. But what happens is you continue to accumulate that iron. And of course, that iron eventually becomes too much in terms of its storage, the storage in the tissue, and it can actually literally rust in the tissue where it's collecting. It can form things like liver spots on your hands, or in the case of the brain, it can actually cause metallic rust to occur, and we can see this. So here's a theory that we've been working on for a while, working with basic organisms, and that is that death itself occurs at a point where there's so much iron in the body, a catastrophe occurs and the cells die because it's just full of rust. Now, you can see this in little organisms. You can see it in flies, where you can measure the amount of iron in a fly as it gets older. And reliably, from its birth until its death, about 2 to 3 months later, the iron levels go up and it doesn't need this much iron. It reaches a point where it gets so saturated with iron. Now, this happens in mammals too. This is some early work that we did on the levels of iron in the brain of mice as they age. And the iron levels go up, and they keep going up. And as long as the animal lives, it keeps going up. And you can see this, other people have looked at this as well, and they've found it in rats, and they've found it in humans. And they've also found that this accumulation of iron in humans, which can be measured now through using instruments like the MRI, the magnetic resonance imaging, which is used for brain scanning, that you can show that that accumulation of iron gets to a point where it's too much and it causes an impairment. And I'm just flashing up some of the papers. To show you how we approach this in a good research institute like the Florey is that we don't guess. We try to come up with an argument, a strong argument about where would you consider making a drug. And in this case, we even studied worms. Here is a worm which has been dissected into 3 pieces. These animals are 1 millimeter long. So you can see its head, its middle, and its tail. And it's been stained with a dye for iron. And here is that same animal towards the end of its lifespan. So at 12 days, you can see the top, middle, and bottom staining brown for all the iron that's accumulated. Does it need the iron? Not at all. It doesn't use it at all. To the extent that we now have demonstrated, and this will be published later this year, that you can take these worms and treat them with an iron-trapping chemical that removes the iron. Here's the lifespan of normal worms. So to read this graph, on the y-axis, you see the percentage of living worms in this population, and on the x-axis, this side, the days. So the normal animal lives to about 20 days. But you can see how many animals stay alive at any particular day after the beginning of this experiment. By day 22, they're all dead, right? It's reached 0, right? That's the normal life curve of a worm. If you were an actuary and you were selling life insurance to worms, you would find this very useful information. So here's the trick. Can we lower the iron in this worm, in these worms, and do anything to its lifespan? And here's the amazing thing. We did indeed treat them with this iron-lowering drug that I'm going to tell you about more in a second. And we extended the lifespan of the worm so that the average lifespan doubled. This is an impressive result. These animals were actually extremely well. They had less iron in their bodies. They lived longer and they were healthier. So here's the big question then. Does this age-dependent rise in iron contribute to Alzheimer's disease? And with that, I'm going to hand over to Dr. Ayton.
So I joined Ashley's group some years back now, about 10 years, and Ashley had some a hypothesis, an idea that was very compelling and intuitive to me. But it was also very radical and counter-dogma. The idea that iron might be contributing to the progression of Alzheimer's disease and not these other factors that the field had decided upon decades ago were the cause of the disease. And so, given that this was such a radical idea, we faced the problem of trying to convince the rest of the field that maybe we've got another idea that's worth considering. So while iron, the potential role of iron had some very intuitive and quite reasonable, hypotheses for why iron might be contributing to the disease, we lacked the clinical evidence. The evidence that from a person who's suffering from Alzheimer's disease to say that iron is actually contributing to their symptoms. So that's what I embarked upon several years ago to try and understand, could we see the effect of iron on individuals? Now, how do we measure iron in the brain of someone? Now, you can imagine this is a very difficult thing to do. We can't, for instance, take a liver biopsy or measure iron in the blood to determine how much iron there is in the brain. But we can do this, and we've applied 3 different approaches. We can look at iron in people who have died, so we can measure iron directly in their brain post-mortem. We can also take the fluid that surrounds their brain, this is the CSF, and we can also now use new imaging technology based on MRI to measure iron in the brain. And I'll show you evidence from all those 3 approaches. But first, before I go into that, I just wanted to talk a little bit about what we now know about the natural progression of Alzheimer's disease. This has been a really, shocking discovery over the last 10 years, that we've found that Alzheimer's disease really is a slowly progressing disease. This is data that's come out of Melbourne under the work by Colin Masters, and this is the natural time frame of Alzheimer's disease. You can see here is the time where people have got Alzheimer's disease symptoms. But before they get those symptoms, there's a 20-year build-up of the pathology of Alzheimer's disease. It's a very slowly progressing disease. And so this would suggest that there are people who are walking around in the community who are quite healthy, but have this disease process occurring in their brain for up to 20 years before they get a diagnosis of the disease. And in fact, we can identify these individuals now by using PET scanning technology, as Ashley referred to. So this is a graph of people who are healthy controlled, ordinary healthy controlled people who've got no memory complaints. These people here have got some memory complaints, we call them mild cognitive impairment, and these people have got Alzheimer's disease. And on the y-axis here, we have the amount of amyloid within their brain as determined by our PET scanning. And what you can see is the really shocking result here is that around 30% of people who are over the age of 65 and are cognitively normal have amyloid in their brain equivalent to that of someone with advanced Alzheimer's disease. Yet they have no cognitive symptoms. It seems that amyloid is not alone sufficient to cause the symptoms of Alzheimer's disease, and there must be something else that might be driving someone getting this disease. So we considered that another factor might be involved is iron. And to look at this, we measured the amount of iron in people who had died and donated their brain, and according to different diagnostic categories. So here is the amount of iron in the brain of people who had died without a clinical diagnosis of Alzheimer's disease, so they were cognitively normal when they died, and they didn't have any pathology within their brain either, and that was confirmed by a pathologist. Here's another group of people who they had a clinical diagnosis of the disease, but they didn't have a pathological diagnosis of the disease, so they were misclassified as Alzheimer's disease. They had a dementia that wasn't Alzheimer's disease. Then there's a group of people who didn't have the clinical diagnosis of Alzheimer's disease, yet the pathologist said that they had the pathology equivalent to that of someone with Alzheimer's disease. So this, again, speaks to the 30% of people who have the amyloid pathology, yet not the symptoms of the disease. But they have the iron level similar to those who are controlled. And then when we looked at the people who had a pathological diagnosis of Alzheimer's disease and a clinical diagnosis of Alzheimer's disease, these are the individuals who had elevated iron within their brain. From a pathology point of view, we can't distinguish between these two groups of people. They've got the same amount of pathology, yet we've got a different amount of iron. So it seems that iron might be driving the progression of the disease from pathology to symptoms. So can we see this in other ways? Well, we took this same group of people, and actually we had clinical information about these individuals in their 12 years prior to their death. You can see here this is a change in their cognition over this time, and this is, the x-axis shows the years prior to their death. All these individuals had amyloid within their brain, and then we separated the groups according to how much iron they had in their brain. Those individuals with elevated iron were progressing in the disease much faster than those with low or moderate amount of iron within their brain. So having high iron in your brain was progressing the symptoms of the disease. We also looked using CSF markers of iron, and so there are factors within the CSF that can tell us how much iron there is in the brain. And I'll just show you one result here. This is, we've taken now everyone who at baseline was considered mild cognitive impairment. So they had some memory impairment, but it wasn't sufficient to cause Alzheimer's disease. Then we assessed the risk of converting to Alzheimer's disease over the next 7 years. And we separated the people at baseline according to how much iron they had in their CSF. And you can see that the risk of progressing towards Alzheimer's disease was much higher if they had more iron within their brain. So this is the lowest amount of iron. This means they've got the least amount of risk of converting to Alzheimer's disease, and the highest amount of iron had the most risk. And again, we could see that this, having high iron, made you get Alzheimer's disease at an earlier time point. Elevated iron led to an earlier onset of Alzheimer's disease. Finally, we looked at MRI measures of brain iron. So there's now a new ability of a new modality of MRI that we can use to measure iron content within the brain. Iron is the MRI is a giant magnet, and of course iron is magnetic, and so we can use these properties of an MRI and of the tissue to determine the iron content in the brain. And this is an example of a scan that we can get where we can measure the brain iron content. So again, we took everyone in a cohort of people who had amyloid within their brain. And then we separated these people according to how much iron they had in their brain according to MRI. And we could show that if you had high iron within your brain, identified by MRI, you've got a decline in memory over a 72-month period. This here is the memory function on the y-axis, and this is time on the x-axis. You can see that having high iron was associated with deteriorating memory performance. Yet if you had low iron, these individuals maintained stability. So via these different approaches, we can see that you can withstand the progression of Alzheimer's disease or diagnosis of Alzheimer's disease if we can keep the iron levels low within the brain. Conversely, those people who have high iron, these are the ones who seem to progress in the disease. So does iron contribute to Alzheimer's disease? Well, these are all correlative measures. We see that having high iron is associated with progression of Alzheimer's disease. It's not yet causality. How do we get to the question of does iron contribute to the progression of the disease? And that's what Ashley is going to talk about.
The way that I feel about this is with that sort of evidence and showing you that we have drugs that can lower iron safely in animals, that you would try a drug to lower iron in the treatment of Alzheimer's disease in a clinical trial. I would imagine that a lot of scientists looking at this sort of evidence would say, well, you're about as certain as being on the goal line and having a shot on goal. But if you're saying kill the full forward, you might hit the post. So money for this sort of thing doesn't come out of thin air. It's very hard to find. A clinical trial is a very expensive enterprise. And the clinical trial that we wanted to do with the drug that lowers brain iron safely, we estimated to cost about $2.5 million. And that's doing it at mates rates and everything, and done at the Florey. And to get that sort of money, we had to apply for grant funding from federal agencies, such as the National Health and Medical Research Council, and they bought it. They gave us the money. So a peer-reviewed assessment of our science said, yes, this is worth a shot. So we have the money, which is really great. What you're looking at up there is a grant application, and gives you some idea that it's not like I just shoot from the hip and say, oh, I think this is a cool thing to do. You have to go through a very, very tedious and prolonged argument about why this really ought to be done. So as a result, we have funding now to do this study, which is going to start next month and has achieved ethics approval. It's called the 3D study, which stands for deferriprone, the name of the iron-lowering drug to delay dementia. And it was funded by the NHMRC, and the object of the study is to test whether lowering brain iron can stop Alzheimer's disease. We are now setting up all the necessary infrastructure so that anyone who is worried about their memory can contact us and see whether or not it's appropriate for them to volunteer for the trial. It's going to be a big commitment. It's a 12-month study. It requires you to have lots of blood tests, and it requires you to be tested in your performance at repeated intervals over the 12 months. But the potential is that within 12 months, we might not just be able to stop you from deteriorating if you've got authentic Alzheimer's disease. There is a possibility as well that you might improve a little bit. We don't know how far this can go. It's the first time it's ever been tested in in people with Alzheimer's disease. The reason that we went with this particular drug, apart from the very exciting effects on worms and other animals, is that it was tested in Parkinson's disease first, where there's a similar problem of elevated brain iron in a different part of the brain. With another reason why we think these age-related brain diseases might be connected by this mysterious elevation of iron in the brain. And because it worked well in the Parkinson's disease trial, we thought, well, we just have to try it in Alzheimer's disease. That was part of the argument. I've put an email address on the bottom of the slide, which is active now. So if you're interested, you can shoot us an email and we will get back to you. But in the next few weeks, we'll be setting up a website and there'll be more public communication about this. Scott and I will be going out into the community, and we're happy to give your social group a talk. Scott likes to get a little food when he's invited. I wouldn't mind a beer, but not at 11 a.m., maybe after 12. So finally, as a little bit of a hoot, I actually have alluded to this, that it might be possible to slow the aging process altogether. Now, it will take a while before we get to the point where our medical colleagues get excited enough about this approach to think, well, let's try it out in aging itself. It's not out of the question. Eventually that might happen. But first we are tackling these diseases. And so I'm going to end off here, and Scott and I will be happy to answer a few questions. I invite you to keep an eye out on activities at the Florey. Part of our mission, obviously, is to continue to do fundraising, and we're going to be doing a number of public engagement events. The University of Melbourne has committed itself to actually forming a centre for Alzheimer's and dementia research, which will be based at the Florey in partnership with the university. You'll hear more about that over the next month or so. And so the amount of excitement behind our ability to actually do promising breakthrough research on these previously incurable disorders has increased, and the government and the university are throwing their support behind it. So I feel like these are exciting times, and if we have got our predictions correct, I think you're going to see some very interesting changes in the practice of medicine over the next 5 years. So Scott, you want to join me, and we'll stop with the slides here and open up for your questions. Thank you.